any point if you can't hear. Uh, yep, that's great. So, sorry, let's start from the beginning. So, welcome to our embryology session today. So, there are some slides which are from previous OSME tutors, so I've incorporated bits of those, and some from my undergrad as well. So, the purposes of embryology, it's again one of those parts of anatomy and technical preclinical stuff which is quite broad and advanced, and it can seem quite overwhelming. But again, the main point to take away is the basic ideas, and especially understanding certain aspects of embryology helps us understand some of the pathophysiology of congenital diseases, and that's fundamentally the use of embryology, which we will try and focus on. So, in terms of an overview, there's obviously a lot more details within the different systems, but broadly, we'll be covering fetal development, cell death, and apoptosis in terms of remodeling and structural fine-tuning of fetal development as well. The CNS development, heart, and GI tract and reproductive system. But obviously, there is also specific details on a urogenital system as well, which isn't being covered today. Okay, in terms of fetal development, I'm just going to see if I can get a pointer somewhere. Bear with me. Of development. We all know the basic concepts of fertilization. That's exactly when the two gametes fuse together, and so you've got the sperm and the ovum cell, which happens in the oviduct or the fallopian tube, as it's known. And then over the course of the journey from um, actually, a pointer would be really helpful. Just bear with me. See, let me just try once more to see if I can get a pointer. No, never mind. Can everyone see my mouse when it's hovering around something? Yes. Okay, that's great. Okay, then we don't need to fuss about it. So, as essentially the zygote, which is the fertilized form of egg and sperm, travels down the fallopian tube into the uterine cavity, you have the cells sort of dividing rapidly. And each of these cell stages, we give it a specific name, just to differentiate it when we are sort of tracking the progress, which becomes more relevant in sort of IVF stuff, and embryology of pluripotency and stuff. But the key bits you need to know is the morula. So that is the cleavage leading to the 16 to 32 cell. So just the stage after the 8 cell and before the blastocyst, which is the stage afterwards. So there is going to be in between stage over here. And the blastocyst then has cavities forming, and you then get to the sort of differentiated stem cells in terms of trophoblasts, epiblasts, hypoblasts, which are essentially just sort of there is a gradient around which the cells sort of divide. And then between those gradients, you then get the gastrulation, which is the formation of the three sort of final layers of the cell division, from which you get individual organs in our body developing. So we go on to the ectoderm, which forms the outermost layer. We've got the mesoderm, which is the middle layer. And finally, we've got the endoderm as well. And each of these layers are the origins for different organs and organ systems in our body, which you can look at in details, and we might have some slides as well. Okay, and this is just showing the actual fertilization itself and the sort of biochemical level process of it, which is a little bit of a recap, but it was a useful diagram for you guys to have a look at, so it's included. Okay, moving on. So, This is again a better diagram to show the different stages. So morula is the 16th stage and the blastula is the 64th stage. And from the blastula, you form this sort of hollow layer, which then forms the three different germ cell layers, which we were discussing earlier on. Okay. And now, in terms of the development of the fetus itself, as I was saying, you differentiate into the different embryological sort of cells. And over here, this is just highlighting some of these layers. So trophoblast is the outer layer, and this is the one which gets attached to the uterine epithelium in the uterus itself. So this is sort of implantation. This is one of the first bits of the embryo which gets implanted to the uterine epithelium. And then you've got the embryoblast, which is going to be the derivatives from which the embryo itself is formed. So this is in day 6, and then you can see there's rapid progression and development within day 6 to 9. You get further, so sort of the syncytiotrophoblasts and cytotrophoblasts are then help to form parts of the placental tissue, which is obviously essential for maternal and fetal circulation, which will be covered in more depth in reproduction. And within the embryoblasts, then you can see has differentiated further into the epiblast and hypoblast. And essentially, hypo is the lower layer and epi is the more sort of epithelial layer here. Moving on, now we start forming the amniotic cavity. So this will then eventually be replaced with some amniotic fluid. So you've got amnioblasts as well, which produce the amniotic fluid and other essential parts of the amniotic sac. And you've also got the primary yolk cell developing from the sort of bilaminar layer of the original hypoblast over here. So all of these are essentially, don't need to know the nitty-gritty details of it, just remember that you've got the trophoblast, which forms the external sort of trophoblast, further differentiates into the placental tissue later on, and then the embryoblast is what the ball of cell, which originally is used to derive the embryo itself. Okay, and also feel free to ask any questions in the chat or feel free to unmute yourself as well. Okay, and this is just showing further stages of the development. So again, we're seeing as we progress on, we have a bilaminar disc of the layered structure. So there's two sort of hypoblast and epiblast, and there is also differences slightly in the cell types. Over here, obviously, you can see it's the columnar cells which are differentiating, not differentiating, sort of dividing and multiplying. And you've also got the hypoblast, which is the yolk cell from which the yolk cell is developing over here. Now, the primitive streak and the primitive node are the first parts of what will then become the nervous system. So this is one of the first things we start developing in embryos. Now, as you carry on, so from the node, from the primitive node to the streak, you eventually form a neural tube, and we will go on to that later on. So these are already, so this is showing the mesoderm, also the second middle germ layer, and the ectoderm is presumably on the outside. And lastly, so without going into too much details about the exact divisions and overall formation, and there's some very cool videos which you can also tune into if you like. This is a good mind map in terms of remembering the layers of division and cells and the names of the different stages of development in fetal development. So this is useful for your exam. So obviously we start off with the fertilized stage of a zygote, which then forms the 16-cell stage of the blastula, which then subsequently forms the 64-cell layer of germ cells after gastrulation. And over here now you are dividing into the three main sort of germ cell layers, and then you also have the gamete layers as well. And these are showing the derivatives of each of the organ systems, as I was saying earlier on. So this is important to know for your exams. So you will often get tested for these either in MCQs or you could be asked to less likely just to hone in on one of the germ cell layers for your essay questions, but MCQs definitely you will have like an option series of options saying which of these is derived from the endoderm. So knowing these are useful, and obviously there's subdivisions of them as well. Okay, we'll talk a little bit about hydatidiform moles. So it's the molar pregnancies which become clinically quite relevant when you're managing ectopic pregnancies and pregnancies where there is a sky-high beta HCG levels. And so these are essentially, you get two forms. So you've got complete mole and a partial mole. And all it means is abnormal pregnancy resulting from sort of abnormal fertilization of the egg and sperm cell. So in a complete mole, you have one sperm duplicating its X chromosome, forming and displacing the maternal DNA itself. So there is an abnormality in the number of chromosomal cells and the distribution of them in terms of maternal and paternal ratios. Whereas partial mole is when you have two sperms fertilizing the same ovum. So the difference is that you just double up the materials ish and then displace some of it. So both of these are not non-viable pregnancies, and often you will see sort of natural processes causing spontaneous miscarriages of these. And if not, if it's too aggressive and if it's part of a sort of choriocarcinoma, which is essentially the carcinoma, cancer of the placenta, then you will often see that those are surgically managed. Okay, any questions so far? No. Okay. And now we're just going to touch quickly on potency and morphogenesis. So morphogenesis, as I was mentioning earlier on, was the gradiental differentiation of your digits and different parts of the body. So essentially, it's turning a blob of an embryo into the defined shape of a fetus at the end of the pregnancy, and that's where morphogenesis comes into play. So it's essentially the use of chemical mediation to allow for programmed cell death in certain parts of the sort of digits to give it a more defined final shape. And that's essentially what we are looking for. So we're essentially programming certain bits of our blob, so to speak, to be eliminated and to form a more defined shape. It's almost like carving sort of woodwork or something. Um, uh, okay. And in terms of potency is essentially referring to how easily your stem cell can differentiate into anything. So, for example, starting from the top, we've got the zygote. So, totipotency, total potency, totally powerful, in other words, to differentiate into any type of cell in the body. Pluripotency is talking about more of you cannot definitely, completely form a complete new human being from it, but you can obviously use it as stem cells to grow organs and even parts of organ systems to form organ system itself. Multipotency, this one's you're talking about sort of specific tissues that can be derived from these stem cells. So you've got adult stem cells here. And so each of these have already differentiated into the germal layers, losing some of its potency. And now they can only differentiate into the fine-tuned tissue that they are stem cells of, rather than an entire organ system itself. And finally, you've got unipotency. So these are sort of the ability to clone one type of cell. So we've got red blood cells here, we've got some gut cells and neurons. So these ones are only capable of differentiating into other neuron cells rather than an entire a few other nervous system tissue and cells. So and this is a little bit more about the chemical patterning I was saying, and limb development also depend on a host of genes working together in order to facilitate those chemical gradients and to allow for the formation of functioning limbs. And again, mutations in any of these genes and any of these cascades that these gene expressions control can result in defects. Okay, well, now we're moving on to nervous system development. Any questions so far? Do we need to learn those genes? Good questions. No, those are purely for entertainment purposes and raising awareness. Just appreciate that it's a complex process. That's all you need to take away from that slide. Okay, nervous system development. So, as we mentioned, we've got the primitive streak, the primitive node, which then eventually developed the neural plate, the neural groove, and eventually the very basic uh, spinal cord and central nervous system. And we can see the several stages, and again, some of it is done through gradient, sort of gene expression controlling some form of biochemical gradients in the most simplest terms in order to result in this development and further specialized structure. So obviously at beginning you've got literally a streak, you've got a node, which then sort of start forming grooves and the plates on either end, and then they eventually form a tube with a cranial, a top, and a caudal bottom sort of hole. And that is exactly what's happening. And this process is called neurulation. Neural patterning is again controlled by a host of genes. And you obviously have the neural crest cells, which have its own form of uh, multipotency um, to then form the layers of nervous tissue. And then you sort of specialize, subspecialize into somites, spinal nerves, and vertebral body. Somites being just neural glial cells, so associated cells of the nervous system. Okay. And this is talking more about the neurulation pattern. This is showing you in 3D. It's a 3D representation, 2D in two dimensions, about how the so this is the notochordus, also the neural primitive streak, and it's essentially there is a difference in concentration of chemical gradient, which pulls the bottom notochord towards the bottom, resulting in the convergence of the either side of the neural plate, resulting in the formation of this tube, so pulling it together, as you can see. And again, this is talking about the morphogens, which result in the formation of this, facilitate this tubal formation. Now, again, these diagrams are way more detailed than what you need to know. Just to appreciate how it comes together and the fact that there is a series of genes and signaling cascades involved in ensuring the precision of this tube formation. Okay, and of course, the reason we learn embryology is to find out what happens if things go wrong. So, one of the biggest consequences of impaired neural tube closure, which eventually obviously closes, is spina bifida. And there are several different forms of it. It's very rare. However, it is one of those congenital defects which have to be corrected as soon as possible. Once either sometimes they're detected antenatally, so patients are typically in theater and beds are booked within the 24 hours of birth. In other terms, if there hasn't been pregnancies where patients were involved with a lot of antenatal care, then you do often see it at birth, and in which case it's an emergency to make sure those patients are seen in theatre as soon as possible. So this is essentially showing what goes wrong. So a gap in the closure of the cord results in herniation essentially of the brain matter. Now you can get different types. You can get the meningeal layers sort of bulging out, forming a meningocele. You can also have myelomeningocele, which is essentially sort of nerves alongside just the meninges. In terms of management of both, it's more or less, same. It's not treated any differently. You will rush these patients into theatre as soon as possible, regardless. Okay, and this is just showing a little bit more about the differences in how they sort of manifest on a diagrammatic level. Um, you can spend your own time in this if you're very interested, but this is just to show you that. Um, so the extent of the presentation is really dependent on how big of a defect there was in the closure of the neural tube. Okay, be moving on to heart. Any questions so far? Okay, I'll have a look in the chat again later. So the next big organ system that we're going to discuss is the heart. Now, obviously, the fetal blood system, circulatory system, and the adult circulatory system, there are some notable differences, purely because the fetal circulation is facilitated through the placenta antenatally, and then obviously once the children are born, um, neonates are born, they then activate their lungs, and then they start the sort of pulmonary circulation like normal adults do. So there are certain changes in the tubing, if you like, that enables these changes in from a fetus to a newborn. So before birth, you have the umbilical vessels, which supply the fetus and take away waste products from the fetus through the maternal circulation via the placenta. You've got the ductus venosus, the foraminal valley, and ductus arteriosus, which again, if we're going to have a look at the fetal circulation in a bit, which is going to show you where these individual ducts are. And all of these are in an ideal case closed, resulting in these remnant structures. However, in cases when that is not happening, is when we have issues. So just before we start talking about those defects, this is a good schematic representation of how the heart develops over the antenatal period. So you've got the very, again, sort of similar primitive blood vessels, almost like a similar to the neural nervous system, of like a cord kind of tube kind of thing, structure, which then starts forming the arterial and ventral. So as you can see, the deoxygenated primitive atrium is sort of on the bottom, and we've got the truncus arteriosus, which are essentially going to then develop into the pulmonary aortic vessels. Um, and again, so as we see the heart taking more of its shape, we can see how the structures differentiate into our modern equipment. So the truncus arteriosus eventually will become the aortic arch, and we've got the venous supply sinus venosus over here, which is going to be the IVC, SVC, and we've got the chambers forming. And we have some important looping and septations. So septations are essentially formation of the final four chambers. And we've got obviously a divide between the atrium and the ventricles itself. We've got divides further between the two atria and the two ventricles. And then we've obviously got sort of demarcated areas for the outflow tracts. Outflow. Okay. Again, this is a diagram to your head around. And again, you do not need to know, every single detail of this or be able to draw it. It is useful to know the structures and what they are going to be eventually developing into. But it's obviously small print in compared to actually learning about the defects of the congenital heart disease. Okay. Now this is, hopefully I have another diagram here. Yeah. So this is now showing in comparison between the fetal circulatory system and the adult circulatory system. So this is just a schematic sketch. We know that the right side has denated blood, the left side has oxygenated know, they start along this way, as demonstrated. Now, needle circulation, we can see a few things differently here. We can see that we've got the ductus arteriosus, which essentially enables us to bypass the lungs and sort of go straight into the aorta. Because obviously in the fetal circulation, the lungs are inactive. So in order to oxygenate the blood, we will need to make sure that it's more taken from the supply of the maternal circulation via the placenta and umbilical veins and arteries. So that's a key difference. And we've also got the foramen ovale, which is right over here, which is the bypass of the two atria here. Now, moving on. So what happens when some of these things go wrong? Any questions so far? Actually, I haven't seen the chat for a while. Okay, none. So, in terms of these ducts, if they do not close on time and with birth, we then start getting a series of problems with newborn circulation and something we know as a group of diseases known as congenital heart disease. So, by birth, we expect the ductus arteriosus to be closed and the foramen ovale closed to form the fossa ovale. However, um, when this does not happen, you get a condition called teratology of Fallot. And this is essentially a combination of um, four defects which result in this presentation. So first of all, you've got a patent ductus arteriosus. So this has clearly not, this duct is not closed, resulting in sort of mixing of oxygenated and deoxygenated blood. Secondly, as a result of that, we've got a increased pressure in our right ventricles, which results in right ventricular hypertrophy, which is the fourth one over here. We've also got VSD. So why do we have that? Again, it's due to the impaired septation early on as well. So this is one of the ductus arteriosus is a presentation of teratology of Fallot, but the origin of this defect results much earlier on in development of the heart when we essentially talk about the septation of the different chambers and the different vessels. So that's when things go wrong. And as a result of that, you also have the aorta directly above the ventral septal defect. Resulting in what we know as an overriding aorta. It's just above the ventral septal defect. Now, all of these, so in terms of, this is a quick note on management. So, whenever, and these result in let me get this right to left shunt. And these are known as a class of diseases called cyanotic diseases. So usually these patients develop with these patients will turn out very blue. You'll have blue babies, as they like to call it. And this results in, prompt investigation of whether it could be tough. And one of the things you manage is you try and decrease the prostaglandin. So you give something called endomethacin to keep this, to make sure that you block. So prostaglandin is what keeps these ducts open. So you try and decrease the level of prostaglandin in the fetal circulation in order to promote the closure of the ductus arteriosus. Okay, that was a quick sort of recap of the congenital heart diseases. Obviously, there's a few others. There's patent ductus arteriosus. Specifically, there is also pulmonary stenosis and so on and so forth. But for the why do you give to decrease? What do you give? So that is endomethacin. I can write that down. It's essentially a prostaglandin antagonist, also an NSAID. Okay, no worries. So the reason why I'm covering only teratology of Fallot is it's the key congenital heart disease that at this stage you should be aware of. There are obviously others you will cover in due course. Some of them are just specifically these defects itself. So ventral septal defect itself is a congenital defect. So is atrial septal defect, which is not covered here. And then pulmonary stenosis being another one. And then you've got the transposition of great vessels, but those ones are not as important as knowing the teratology of flow. So that's why this is the only slide we've got on congenital heart disease. Now, moving on to GI tract development. Now, the GI tract development is very complex, but the principles of it is very similar to the nervous system and the nervous system. So you start off with sort of primitive stages of the embryological tissue, and then you sort of then become more differentiated and specialized. For the purposes of time, I'm just going to talk about the defects associated with um, the development of the GI tract. So one of them is Hirschsprung's disease, and this is essentially uh, due to the lack of sensory innovation and afferent innovation to the gut. So when you lose those innovations, then you lose the ability to facilitate peristalsis in your gut, which results in, fecal impaction, not moving your gut. So constipation, there is a delayed passing of meconium, which is essentially the first fetal sort of feces. And all of these can then be used to manage these patients quite early on because it can otherwise become quite serious because obviously you have a blockage or you cannot really, it's a very slow sort of traffic system, right? And you're hardly moving any traffic out of it. So you're going to build up more and more of a bottleneck. And that is exactly what happens in our gut as a result of this. And then the other key one is omphalocele and gastroschisis, which are essentially two fancy sort of terms which talk about two set of defects which happens due to impaired fusion of the vitello-uterine duct. I'm just going to show pictures of that somewhere. Okay. So the embryological origin is the vitello-uterine duct, which, because of impaired fusion and differentiation, you result in these defects. Um, and you have a herniation and malrotation of the small intestine itself. Okay, so you've got rapid growth, and then you sort of rotate the wrong way because of the impaired fusion of these ducts. And this results in something called an omphalocele or a gastroschisis. Okay, I haven't covered gastroschisis here, but essentially, omphalocele is the herniation of the small intestine through your belly button, so umbilicus. And usually, the difference between this and gastroschisis is that omphalocele is covered by a thin membrane, and gastroschisis is literally like a blob of small intestinal tissue, which might not be presenting just directly through your umbilicus. It might be a little slightly deviated or to your side. And this is obviously as soon as the baby comes out, you will spot this defect if the baby is to have any. A lot of the time nowadays, it's also diagnosed antenatally. And obviously, in terms of prognosis and management, it's a little bit more aggressive in terms of management with a gastroschisis, simply because there isn't a covering membrane to trap any moisture loss and fluid loss. So those patients are typically wrapped with cling films and then hurried into theatres as well to correct for the defect and close up the hernia. And obviously, one of the biggest risk factors with omphaloceles, if delayed, then you get malrotation of the gut, and that results in a very poor infant who will be vomiting up and just very look very ill. Okay, and lastly, we are going to cover the genital system. Now, again, big topic, especially because there's two different subdivided reproductive systems. But again, big pictures, broad spectrum concepts. So first of all, we've got the chromosomal determination, right? And so that's determined through the SRY gene. Um, and essentially these flow diagrams, again, you don't need to know details, but it's a good pictorial representation of how we do have the sex determination, right? So we've got, to begin with, we've got the two ducts, of which one of them will then go on to developing to the female reproductive system, which is the Mullerian duct. And we've got the Wolffian duct, which continues to become the male reproductive system. Now, the presence of SRY essentially results in the maintenance of the Wolffian duct, and it produces no anti-Mullerian duct hormone, as a result of which the Mullerian duct itself is disintegrated, resulting in only the Wolffian duct remaining and further developing into the male reproductive system. In the case of the absence of the SRY gene, you produce something called the anti-Mullerian hormone. So this protects against the Mullerian duct from being apoptosed. And as a result of that, you result in the formation of the female reproductive system. Right. Yeah. So this results in the so the Sertoli cells in the male reproductive system as well, further on, to enable a positive feedback loop for the maintenance of the Wolffian duct, um, produces testosterone, which is produced by the Leydig cells, and this allows for mesonephric is another term for Wolffian duct, and the paramesonephric duct is essentially apoptosed through the production of anti-Mullerian hormone through by the Sertoli cells. So in other words, sorry, I need to clarify something. So you do not have any production of anti-Mullerian hormone in this case. Does that make sense? Or have I confused people? Okay, I'll carry on. Um, yep. So this is just showing this is another representation of the individual ducts. Okay, and this is just showing the actual active testosterone component, which facilitates these uh, effects. Okay, and lastly, this is a table which you're not required to learn off by heart. However, what I want you to appreciate from these is these branchial arches. Now, it's just from the head and the neck, you have these sort of structural arches, which are almost like branches, a bit like the brachial plexus, where each of these are designated to um, differentiate into a few of these structures as outlined. And these are useful in terms of looking at origins of defects and where in itself, like in development, did things go wrong in order for a certain defect to manifest. Um, for you guys, sometimes they like asking MCQ questions on which branchial arch does the pharyngeal muscle develop from. For those sort of things, this is a useful table to just remember. But again, this is if you're trying to prioritize on revision, this is going to be quite low yield in terms of and very niche in terms of MCQ. So only prioritize this if you have the time or you're interested. Okay, so that's everything I have. And does anyone have any questions before we move on to MCQ questions. No, not quite. Okay, in that case, what germ layer is the stomach epithelium derived from? Okay, we've got one answer. A, endoderm. From Noor, any other takers on any other advances from anyone agree or disagree with Noor? No? Okay, let's reveal. Is the endoderm? Well done, Noor. So again, it's one of those things where the table, it helps to get used to them and look at the tables to familiarize yourself. And also there's some good videos which shows the different germ layers developing into the different components. But again, it's sort of more of the internal structure in terms of our organ system, so it makes sense that it's one of the endoderm. Well, some of them are a bit counterintuitive, but if you can link it in some way as well, that helps with remembering. Next question. What is the name of the vessel that will close at birth to prevent blood flowing between the aorta and pulmonary circulation? So we've talked briefly about all of them. Any takers? Good. Got a C. Brilliant. That is correct. Well done. Well done, Noor. Okay. Which of the following muscles? Again, so this is a very typical question that you can anticipate as one of your niche MCQs. And again, appreciate I've just given you the table. Um, and also pharyngeal arches and branchial arches are synonymous. Okay, two answers. Lovely. B. Indeed. Muscles of facial expression. Gosh, you guys must have. I think A is from first Dutch, so I'd say B. Uh, Brilliant. Yes, very good. All right answers. Indeed. So again, it's one of those things. I don't know if you've come across those tables before. Otherwise, you guys have very good photo print memory. So well done. Okay, and how does an omphalocele generally form? I feel like we've got a response. Which arch is C from? Okay, we'll come back to that question. Sorry, I've lost the question as well now. So we will come back to that question, Demi, in a second. Any takers on MCQ 4? Well, I've revealed it now. So yeah, so the vitelline duct is the umbilical duct. Vitelline arteries supply the umbilicus, so that's another way you can associate it. Um, okay. And finally, which of these processes, if abnormal, can lead to a hydatidiform mole? Mm, brilliant. It is indeed fertilization. Well done. Okay, so let's go back to the question of which arch is C from? Let's go back to the question as well. Which arch is C from? Pharyngeal constrictor muscles. So that would be the cricopharyngeus. So if we look at our table, so stylopharyngeus would be, I would say, 3. I'm going to double check this as well because I don't want to misguide anyone. But other than that, do we have any other questions while I check the answer to the pharyngeal constrictor muscles? So I was wrong. It's the fourth pharyngeal arch. And why is that? Let's have a look. So yeah, pharyngeal constrictors would be pharyngeal muscles. Um, so it would be the fourth arch. Does the question not make sense in that case? Yeah, so that would be the fourth one because we said the second pharyngeal arch. Does that answer your question, Demi? Uh, Omphalocele is because the abdominal wall do not form. It's not so much of the abdominal walls. Yes, there is a fundamental defect, but it's more of the through the so the omphalocele is through the umbilicus, right? So it's not necessarily a question of the abdominal wall itself. It's more of the herniation of the small intestine through the umbilicus itself because of defects with the umbilical tube, congenital umbilical tube. Does that make sense? Okay, you awesome. Okay, great. But other than that, uh, if there are any more questions, I'm happy to stick around for another few minutes. Otherwise, thank you everyone for your time. Please, which I will put back on the screen and ask any other questions. Ornia, are you still online as well?
Yeah, thank you so much, Nadra. That was great.
Okay, perfect. I'm guessing they filled in the form, so I
I
will
think
leave.
maybe if we leave it up just for a couple more seconds and then we'll wait for everybody to leave, but that should be good.